All right, hi everyone. We are going to talk about mutations today, specifically point mutations, not chromosomal mutations. But before we do that, I want to talk about Beadle and Tatum and the Nobel Prize that they won for the experiment that they did with their bread mold. So they're the ones that came up in the 1950s with the idea of the one gene, one enzyme hypothesis. And at the time, it was groundbreaking. Now we know a lot more information, so we have to modify their hypothesis, which is what science does. So instead of one gene, one enzyme, we now understand that one gene could produce several proteins, meaning, remember, when the mRNA gets made, the introns get removed and the exons get put together, and different exons can be put together in different ways. Alternative splicing, that's what that's called. And so we can put it together and produce different proteins. So we have one gene, a few proteins that come from it, but we also know now, so only about 1.5% of all of our DNA actually codes for our proteins or enzymes. About 75% of our DNA, we have now discovered, only codes for mRNA. So it just gets transcribed, but it doesn't get translated into proteins. And we know that those mRNAs, their job is to regulate or control gene expression. So turning genes on and off, that's their job. So about 75% of our DNA, that's what we think right now at least, is its job is just controlling the DNA and which genes are being activated. But let's go ahead and take a look at Beadle and Tatum's experiment. Remember that the College Board is focusing on experimental design during AP Biology, and so most of your AP exam will be about experiments and you analyzing the experiments. So the more exposure you have to experimental design, the better off you are. So let's go ahead. I'm going to skip this and talk about what they did. And I might scooch it over just a little bit if I can. These prezzies are awkward to work with. Okay, so they started with a type of bread mold that they could grow, and it's called the wild type. Wild type, you guys, means normal. It's like a non-judgmental way of saying what is normally present in the organism, and then mutations are not normal. So they're not the wild types in that case. So they started with some wild type bread mold. The reason they chose the bread mold, it was very easy to work with. They are haploid organisms. I'm not going to go into the details, but just trust me that it was an easy organism for them to work with. And they discovered that they could grow the bread mold on this particular food source that we're referring to as the minimal medium. And so they're calling it minimal because it hasn't been supplemented with anything. We're going to supplement it in just a minute. But the bread mold can grow on this minimal medium. They then zapped the bread molds with x-rays, and we know, and they knew, that x-rays damage DNA. So they wanted to see what would happen to the bread mold as they zapped it with x-rays, what parts of the physiology of the bread mold would stop working. So they focused on a very specific pathway, and this is the pathway right here. I'm going to show it over on the side here. This is a pathway that forms the amino acid that's called arginine. Arginine is one of the 20 amino acids that we need in order to build our proteins. And this is the pathway for how the bread mold made their own arginine. They started with a precursor molecule. Now, that's a word that you're going to see in AP Biology a lot, and you need to understand the prefix pre means before. So this is the molecule that the bread mold is starting with, and then its enzymes are going to keep adjusting the molecule until it gets the target molecule, which is an amino acid called arginine. So this is the arginine pathway that they're studying. If the X-rays damage the DNA of other pathways in the bread mold, that's not what they were studying. So they got ignored. They were just focusing on which bread molds had damage in their arginine pathways. And then they started to classify those different mutations. So what we know about the steps of the arginine pathway, we start with a precursor molecule. There's an initial enzyme called enzyme A that adjusts it and turns it into another amino acid that's called ornithine. Ornithine is not one of the 20 amino acids that we use to make proteins. It is an amino acid, but it's not used to make proteins. So you won't find it on the genetic code chart of all the mRNA codons. So it's not good enough. So then the bread mold has another enzyme called enzyme B that converts ornithine into citrulline. yet another amino acid that we don't use for building proteins. And then enzyme C converts citrulline into arginine. And that's the ticket. The bread mold and you and I absolutely have to have arginine in order to build our proteins. It's like trying to write in English without the letter E if we don't have arginine. We can't write words without the letter E, and you can't build your proteins without arginine. So if there's damage in the arginine pathway, then the organism, the bread mold, can't grow. It can't make its proteins. It dies. It's not able to grow. So then they started to study, well, what if we supplement the diet of these bread molds? Could we get them to survive? So they categorized these mutants into 3 different categories. So let's take a look at them. The class 1 mutants, they can't grow on the normal medium because something's broken in their pathway. However, if we add ornithine, citrulline, or arginine to their food, they suddenly grow just fine. So the scientists, Beadle and Tatum, figured out, and let's see if I can scooch this over, exactly what was wrong with these class 1 mutants. They had damage in their DNA that caused enzyme A to no longer function. And so their precursor molecules, they had their precursor molecules available in their food, but they couldn't convert it to ornithine. So the entire process was broken. And so they couldn't make arginine, they couldn't make their proteins, they died. However, when they supplemented the diet with ornithine, the enzyme B and the enzyme C were still working. And so they were able to go through the rest of the process and produce arginine. Same thing if they were given citrulline or arginine itself, they did great. So then the class 2 mutants, let's scooch it over. The class 2 mutants, they can't grow on normal food, but they can't grow in ornithine plus the food either. That's not good enough. So but if you give them citrulline or arginine, then they're able to make their proteins and survive. So the scientists, Beadle and Tatum, figured out that this time the X-rays must have damaged the DNA that made the enzyme B. And so they have the precursor in their food, these molds, the food has the precursor. They have a functioning enzyme A that allows them to make ornithine, so they're on the right path, but then enzyme B that converts ornithine to citrulline doesn't function. So then they cannot finish this pathway on their own. However, if we feed them citrulline, they bypass this broken enzyme, and then they have enzyme C and they can make arginine and they survive. You can guess where the last class of mutants were. The last class of mutants, they cannot survive on regular food. They can't survive on supplemented with ornithine or citrulline. The only way they can survive is if you give them arginine directly. So if you go back to the class 3 mutants, they have the precursor molecule available in their food. They have enzyme A that is functioning, so they can make ornithine all by themselves. They have a functioning B, so they can make citrulline all by themselves. But at the very end of this pathway, they can never convert citrulline to arginine, so they can never make arginine. And so they cannot survive with any amount of supplementation except for arginine itself. If you give them arginine, then they can survive. So this is where Beadle and Tatum came up with their 1 gene, 1 enzyme hypothesis. It's an amazing experiment. We've learned so much since then. All right, so let's jump ahead. Give me a second. There we go. So now we're going to talk about our main topic today, which is mutations. And these are pretty straightforward. We have already talked about chromosomal mutations. That's where we had, for example, deletions of an entire section of a chromosome. That might involve thousands of nucleotides that are gone at one time. What we're talking about today is point mutations, where maybe 1 letter of our 3 billion letters gets changed, and that has an impact. Sickle cell anemia, cystic fibrosis are both genetic disorders that you've probably heard of. I know you studied sickle cell anemia already. 1 letter out of 3 billion got changed, and that was enough to create those genetic disorders. So those are going to be called point mutations, and that's what we're going to talk about now. So it says, it becomes clear how changes in DNA can affect changes in protein structure and physiology. There are 2 major types of DNA level mutations, not chromosomal mutations. That's where we deleted an entire section or we had inversions or translocations. That was from a little while ago. That's chromosomal level mutations. DNA level is at the molecular level. We have some sort of a change. All right. So, and then this is an example of sickle cell anemia. Here are 3 of the letters in the hemoglobin of wild type. So we're not judging by saying normal. We're saying wild type. So this is the wild type. And then in this case, an A instead of a T and a T instead of an A. So it produces an mRNA that has 1 letter wrong. Now, keep in mind the idea of wobble. If that letter had been the 3rd letter instead of the 2nd letter, we might not have had this mutation. So if 1 letter had changed and instead this A were a G, we would still have glutamine here and it wouldn't be a problem. But this 1 letter changed and caused a different amino acid to form there. Now, I want to bring you all the way back to your biochemistry to make you remember why one amino acid could make a huge difference. Remember that amino acids have an amine group on one end, NH2 or NH3. They have a carboxyl acid group on the other side, and then they have the part that makes each one unique called the R group. And remember that each R group has its own chemistry. So some of them were polar and some of them were nonpolar. Some of them had sulfur. Some of them were attracted to water, not attracted to water, all kinds of things. So that's what determined the folding of the protein. It turns out that valine has a different enough chemical identity, that it causes the hemoglobin to fold differently and leads to then sickle cell anemia. All right. So let's look at the different types of point mutations. The first type of mutation, which we've already talked about, just making sure we're clear on it, is called a silent mutation. And that's where 1 letter gets changed, usually the 3rd letter in a codon, and it still codes for the same amino acid. And so there's no phenotypic change. The next one is called missense. Missense is when 1 letter gets changed again, and we get a different amino acid. So that's what happens in the case of sickle cell anemia or cystic fibrosis. The next type is called nonsense. And I think it's super easy to mix up nonsense with missense. So I don't know if this will help you, but the way I always remember it is no nonsense. Stop your nonsense. Nonsense mutations lead to stop signals. So premature stop codons that stop the formation of the protein. So notice that this particular protein was supposed to be going and it comes to a dead stop because of that. I want you thinking in the back of your head about the location of the point mutations and how that totally makes a difference. What if the mutation, instead of being right at the beginning, what if that mutation was all the way at the end? How would that have affected the structure of the protein? It's dramatically changed at the beginning of the protein. Later in the protein, it might not have been as much damage. Okay, the next thing that we want to talk about are frame shift mutations. And I'm going to use, I always like to do this. I feel like this is a good way to show students frame shift mutations. So I'm going to jump over to another screen. Bob the Fat Cat ate. Can I move this a little bit? I'm trying to, there we go. Bob the Fat Cat ate. Whoops, I don't need to start that. Okay, so what I have here is I made a stupid sentence that just involves words that are 3 letters long. Your mRNA is read by your cell machinery, your ribosomes, 3 letters at a time. We'll call those codons. So it's similar to a sentence that is that has words that are 3 letters long. So Bob the Fat Cat ate. Now, in a frame shift mutation, well, let's back up. Let's do just a point mutation like we've been looking at. So with a point mutation, let's change Bob to Boz. Boz the Fat Cat ate. With 1 letter changed, it's possible that we can still know what the sentence means. We pretty much get the idea that a fat cat named something ate. And that's the same thing that happens with most point mutations. The protein is mostly still okay. There might be a little bit of problem with the protein, but it's mostly okay. So that's with a substitution. That's where I substituted 1 letter for another. But now let's try something different. Let's try the insertion or deletion of a letter. So let's just add the letter Z here. We've inserted it instead of substituting it. Now remember that the ribosome can only read 3 letters at a time. So let's change this so we're reading 3 letters at a time. So give me a second. We're going to read, 3 letters at a time. This is what an insertion does to the sentence. Every single codon after the insertion, so here's the insertion, the Z is the insertion, every single codon after that is ruined. And so every single amino acid is ruined, and then the entire after, so they call it downstream of the mutation, everything is ruined. Let's change this, and instead of, we'll go back, I think we could do it this way, couldn't we? Instead of an insertion, let's do a deletion. So let's delete the letter T this time. Same concept. We got to read it in groups of 3. So I'm remaking my groups of 3. These represent our codons. Notice how badly damaged the sentence is. When we had a substitution, we just traded one letter for another. We still got the gist of the sentence. But once we insert or delete a letter, we throw off what are called the frames, the groups of 3 letters at a time. And so the frames are off. And so this is called a frame shift mutation. All right, so let's go back to talking about our frame shift mutations. Give me just a second here. All right. So the first type of frame shift mutation is called extensive missense. This is horrifying when it says it's extensive. The reading frame of the ribosome is altered so that all amino acids downstream from the indel means insertion or deletion, it doesn't matter which one, they're all altered. So the earlier that we have a frame shift, the more damage it does. If it's later in the gene, then it's not as bad. Immediate nonsense, the reading of the frame of the ribosome is altered so that a stop codon is introduced prematurely. So remember, no more nonsense. Stop that nonsense. That's a stop sign. And then limited effect, hopefully you're thinking this through. If I inserted letters 1 or 2 letters at a time, I totally screw up the framing. But what if I insert or delete groups of 3 letters at a time? That's called limited effect. If the change is in a multiple of 3, I'm still going to get, so maybe I removed the word the. So now it's Bob Fat Cat ate. It's still the correct amino acids. It's just that one of the amino acids or 2 of the amino acids, if it's in groups of 3, have been removed or added. And so it's less effect that way. All right, ladies and gentlemen, that's it.